First thing to say is that it was my pleasure to be presenting an update of the ASCEND-CML study on behalf of my colleagues and co-investigators at the Australasian Leukemia and Lymphoma group. This is a trial where we use Asciminib for newly diagnosed chronic phase chronic myeloid leukemia patients. And it was one of two studies for Asciminib in the frontline chronic phase CML setting. By way of background, Asciminib is a drug now approved in many countries for third-line indication for chronic phase CML, and it's just been approved for treatment-naive chronic phase CML in the US on the basis of another study called ASC4FIRST. We are also one of the first studies to report outcomes alongside ASC4FIRST for frontline chronic phase CML. Asciminib belongs to a new class of CML inhibitors or BCR-ABL inhibitors called STAMP. It stands for specifically targeting the ABL myristoyl pocket. It acts differently to the other drugs that are on the market, which binds to the ATP binding pocket. Asciminib binds to the myristoyl pocket. We have published the primary analysis already, and at ASH this year we gave an update on our new on our data, which is maturing in terms of efficacy as well as safety. The first thing to say is that the deep molecular response continues to climb. So, for instance, the MR4, which was initially reported to be about 50% at 12 months, we now got data out to a median of 36 months with a minimum follow-up of 24 months. So the MR4 was 50% at 12 months. At 36 months is close to 73%. The MR4.5 rate at 12 months was 31%, now is about 55% at 36 months. So the deep molecular responses continue to climb, and that's really exciting because it means that we can get more people to a trial of treatment-free remission with the intention of stopping therapy. The second thing to say is that in terms of adverse events, we saw a number of hematological toxicities such as neutropenia, anemia, and thrombocytopenia, but they were mainly observed in the first year of treatment. When patients get into the second and third year, there were very little in terms of events reported. Similarly, the most common biochemical abnormalities such as lipase and amylase elevations, they were mainly reported in year 1 and much less so in year 2 and year 3. The other favorable safety feature that we observed was that there was only one vascular event reported so far. It's the same as the patient that we reported last year, having 2 transient ischemic attacks, making full neurological recovery. We've only had 1 blast crisis, which was a lymphoid blast crisis. Again, that was reported last year. So very favorable safety profile and continuing accrual of deep molecular responses. Now, on the other hand, we did see a number of patients, a minor minority number of patients that developed treatment resistance. And the pattern of treatment resistance that we observed was that the resistance mechanism was conferred by myristoyl pocket mutations as well as kinase domain mutations, where 8 patients lose response, and 5 of the 8 have mutations at the A337 residue in the myristoyl pocket of ABL. 3 of the 5 had M244V, and 1 patient had a T315I in conjunction with a 244V. So what we're seeing is that the majority of patients who do have treatment resistance are losing their response because of myristoyl pocket mutations, and these mutations mean that they're salvageable with other drugs such as dasatinib, which are already on the market. But it also speaks to the fact that the agent is very, very specific and inhibits BCR-ABL right on point. And the way that the disease escapes is to actually mutate residues in the ABL myristoyl pocket. So I think that's probably the most pertinent update. And we're continuing to accrue data on this ASCEND-CML study whilst we get the next study off the ground, where we're going to combine Asciminib with dasatinib for patients with high-risk chronic phase CML as identified by additional mutations in genes such as ASXL1, RUNX1, and DNMT3A.